We're exciting for Friends Annual Meeting 2024. Let's dive into Session 3. In Session 3, we'll discuss an aligned strategy to integrate ctDNA into oncology clinical trials for use as an intermediate endpoint. Intermediate endpoints are important to support accelerated approval. Circulating tumor DNA, or ctDNA, is a molecule found in the blood that is thought to indicate solid tumor burden. To use ctDNA as an intermediate endpoint in regulatory decision-making, FDA expects aggregate meta-analyses at the patient and trial level. Over the past few years, Friends CT Monitor project has performed a series of aggregate patient-level analyses demonstrating a decrease in ctDNA associates with improved outcomes, including overall survival. We'll have another data readout as part of the annual meeting focused on advanced non-small cell lung cancer treated with an anti-PD-L1 and/or chemotherapy. In addition to developing data to support using ctDNA as an intermediate endpoint over time, Friends also created a roadmap to support which data need to be developed. This document outlined 3 key areas: technology considerations, including assay types and validation strategies, clinical considerations, including clinical contextualization, and sampling considerations like timing and frequency of collection. Our next step is to align on key outputs for prospectively designed trials. During the Friends annual meeting, we'll discuss an aligned strategy for integrating ctDNA into prospectively designed trials. The working group is also working on a white paper that will be released before the meeting, which we encourage you to read before you attend. The objective of this work is to align on standardized, harmonized, and robust data collection to include in prospectively designed trials that can support more robust meta-analyses. We outline various aspects of a prospective trial that incorporates ctDNA, including assay characteristics, timing of sample collection, patient inclusion considerations, calculating a molecular response, approaches for comparing treatment groups using molecular response, and meta-analysis considerations. During the annual meeting, Session 3, we'll begin with a data presentation of CT Monitor findings. Then the panelists who are outlined on the right of this slide will discuss an aligned approach for incorporating ctDNA into prospectively designed clinical trials, focused on using ctDNA in advanced non-small cell lung cancer treated with immunotherapy. They'll also review considerations for a meta-analysis approach that includes outstanding questions. Please register for the meeting using the QR code on the right or visit our website at friendsofcancerresearch.org. We look forward to seeing you on November 12th.